Okay, so we're talking about the lysosomes. We've talked about them being involved in in degradation of old or damaged organelles. These are rich in hydrolytic enzymes, so they contain proteases, they contain nucleases and lipases. So for example, the lysosomes can be used in autolysis. For example, during the metamorphosis, the tadpole for that tail to be reabsorbed, the lysosomes are needed for that. So those are the lysosomes. The other organelles that we have include what are known as the peroxomes. The peroxomes. So these have a single membrane, and they actually originate as buds from the endoplasmic reticulum. They originate from the endoplasmic reticulum, the peroxome. So this is the one in blue, depicts a peroxome. So these are sites where reduction and oxidation reactions take place. The redox reactions will take place in the peroxomes. So for example, the peroxomes that are found in the liver, they contain enzymes that are able to remove electrons from or oxidize the ethanol in alcoholic beverages. So people that drink, someone drinks, these peroxomes will be involved in the removal of electrons or in oxidation or in oxidizing the ethanol in alcoholic beverages. So these, in other words, we're saying the peroxomes contain digestive and detoxifying enzymes. And they are called peroxomes because the digestion and detoxification process leads to the production of hydrogen peroxide as a byproduct. So that's the function of the peroxomes, detoxification and digestion. They are involved in the redox reactions, reduction and oxidation reactions. We have the vacuole. The vacuole. So when we look at, especially when we look at the plant cells, they have these specialized membrane-bound structures that are known as the vacuole. So usually the vacuole will have a membrane that is called the tonoplast. So the tonoplast contains channels for water, which are used to maintain the tonicity or osmotic balance of the cell. This membrane, the tonoplast, is used for the maintenance of the tonicity of the cell. So target pressure is maintained by the vacuole. And then the other function is that a central vacuole is also involved in cell growth. How is that involved in the cell growth? Because it occupies most of the volume of the cell. So plant cells are able to grow by expanding the vacuole rather than increasing the cytoplasm. So the vacuole is going to expand and the plant cell is going to grow. So they're also used for growth as well. That's the vacuole. So the vacuole is present in all plant cells and also fungal cells. Then we also have the protozoa, some protozoas and some animal cells we also have the vacuole. So the function of it is that it's able to store water, it's able to control the target pressure, as I mentioned. It's also able to control the pH of the cell. And it is also involved in the removal of toxic or waste materials. So it's used for storage of water, it's used for controlling the target pressure, it's used for controlling the pH of the cell, and also for removal of toxic materials. Then the other part organelle that we have is the mitochondria. The mitochondria. And when you look at the mitochondria and the chloroplast, these are actually share similarities in their structure and functions as well. Structurally, they both contain a double membrane. And then, interesting enough, they also both contain their own DNA and protein synthesis machinery. Functionally, these are both involved in energy metabolism. Remember, mitochondria metabolize a sugar which generates ATP. And then when you look at chloroplast, is involved in the process of photosynthesis. So these are similar. When you look at the mitochondria, it's present in all eukaryotic cells and has two layers of the membrane, as I have mentioned. So you have the inner layer, you have the outer layer as well. The inner layer is involved in the production of ATP during cellular respiration. And then we have the cristae. We have the cristae, which has two compartments. We have the matrix. There's a matrix compartment, and also an outer membrane or intermembrane space. That. So now, on the surface of the inner membrane, and also embedded within it are proteins that carry out oxidative metabolism. Those proteins are going to carry out oxidative metabolism. That same process for ATP production. And then remember I mentioned that the mitochondria have their own DNA, which contains various or several genes which produce the proteins that are essential for the function of the mitochondria. However, we find that these mitochondria are actually not fully autonomous, because we find that the genes that encode the enzymes that are used in the metabolism here are actually located in the nucleus, in the DNA of the nucleus. The outer membrane, mitochondrial membrane, is smooth and is able to allow many small molecules to pass through it. However, when we look at the inner membrane, as we have seen, the inner membrane has numerous folds, and this strictly regulates the types of molecules that can move across it. These folds are what are known as a cristae. So crista is just one fold. Cristae, it's a main fold. This cristae will be extending into the matrix. Now, what is the function of cristae? The cristae are going to increase the surface area of the inner mitochondrial membrane, which provide the surface of the chemical reaction to transform the energy in food into ATP. So it increases the surface area. And then we have the chloroplast. The chloroplasts are present in plant leaves and also in some algae that undergo photosynthesis. It is also surrounded by two membranes. So you have the outer membrane and the inner membrane. So it's rich in hydrolytic, or rather, the inner membrane is rich in chlorophyll and some other pigments as well. It also contains enzymes and electron carriers. Remember, it's similar, some similar functions to mitochondria. So the chloroplast also contains enzymes and also electron carriers. These membranes are the sites where photosynthesis takes place. So they are parts where photosynthesis takes place. So chloroplast contains chlorophyll, which traps light energy for photosynthesis. Another interesting thing is that chloroplasts also contain a variety of light-absorbing yellow and orange pigments that are known as the carotenoids. Carotenoids. If you remember when you looked at lipids, we mentioned the carotenoids. So these are contained in the chloroplast. The chloroplasts have a closed compartment or have closed compartments of stackled membranes, which we call the grana. So this is a granum, which is singular. Plural is grana. So this grana will lie inside the inner membrane. And then it also have it also has the stroma. Now the enzymes that are used in the synthesis of glucose during photosynthesis are actually contained in the stroma. And then a chloroplast can actually contain one or more grana. Now each granum may contain from a few to several dozen disc-shaped structures that we call the tharacnoids. The tharacnoids. They can contain the tharacnoid. So now on the tharacnoid, on the surface of the tharacnoid, are the light capturing photosynthetic pigments. So the light capturing photosynthetic pigments are found on the tharacnoid. So that's the chloroplast. So remember also the chloroplast also contain DNA. However, many of the components for the chloroplast are actually located, many of the genes are located in the nucleus. However, we find also that some of the elements, however, that are used in photosynthesis are entirely synthesized in the chloroplast itself because it contains DNA. And then we have the cytoskeletons. We have parts what are known as the cytoskeletons. The cytoskeleton structures. So these are major structural features. The cytoskeleton. They are major structural features and are common to all eukaryotic cells. They are an extensive system of protein fibers. So what they do is that they give shape to the cell and also structural stability. They give structural stability and shape to the cell. Additionally, cytoskeletal proteins are involved in moving the cell itself and also to move materials within the cell. So what am I saying? We are saying, in addition to giving the cell its shape and also its structural stability, these cytoskeletal proteins are involved in the movement of the cell and also in the movement of materials within the cell. So in essence, we are saying these cytoskeletal fibers organize all the organelles and other cellular structures into a cohesive whole as a whole. So the eukaryotic cells may contain 3 types of cytoskeletal fibers. So you have the actin filaments, which are sometimes known as the microfilaments. We have the microtubules and the intermediate filaments. Microactin, microtubules, intermediate filaments. These are the cytoskeletal structures. So they're involved in movement, They're involved in feeding, they can be involved in sensation. For example, organisms that contain cytoplasmic projection like the cilia. So we have cells that contain cilia, we have cells that contain flagella. These are going to contain these proteins, the actin, the microtubule, and the intermediate filaments. And they're going to use these for moving, for feeding, and for sensation as well. So like I mentioned earlier also is that these are also used in shape, providing shape and support to the cell. So reproduction in eukaryotic cells can be asexual or sexual reproduction. So the most common form of asexual reproduction is mitosis. So mitosis happens such that the cell is going to grow double its size, and then it's going to divide. So the cell is going to grow double its size, and then it divides to form two identical cells. These daughter cells will be identical. And then we also have budding, the process of budding, which is usually happens in these organisms such as unicellular fungal cells, and also in some protists or protozoas. They can divide via budding. This is budding. Simply a bud comes out and then it separates. And then also binary fission, could be binary or multiple fissions, are also observed in some primitive eukaryotics. And then some fungi are also able to reproduce via sporulation. So these are some of the methods of reproduction in eukaryotics. And then there's also sexual reproduction. So here, the cells of the reproductive system in plants and also in animals are able to reproduce by sexual method. So during this process, what happens is a process of meiosis. So the cells are going to divide meiotically, which will lead to formation of four daughter cells. So four daughter cells, and we are going to look at these in more details when we look at cell division. We're going to look at mitosis and meiosis as well. So that's sexual reproduction, that's asexual reproduction. And those are the functions of the organelles. Those are the structures of the organelles. So having reached to this end, are there any questions? Any questions, guys?
No, no question.